Before we proceed at all, we have to review the three basic concepts of genetics. And some of you will have had biology and some genetics and will know them, but it will help to review these concepts. The first is the notion of information, that information is carried in the sequence of the DNA. In the same way that semantic information is contained in the sequence of letters of an English text, the genome, human or of any other organism, carries the genetic information in the sequence of letters of the DNA alphabet. Second is mutation. The information is transmitted by and large very faithfully from one generation to another, but once in a while there is an error, a typographical error, if you wish, happens. Now, errors in the DNA happen much less frequently than happen in books. You know, the probability of a mutation typically is of the order of 1 in 100 million. That's a very good fidelity. But these mutations are responsible for hereditary diseases. They are also responsible for the possibility of evolution. Mutation is not necessarily a bad process. Without mutation, evolution would have not occurred. We would never have come to be. And then the concept of development, which is very difficult to understand. I meant to say the opposite. It's very easy to understand, but very difficult to incorporate into the mental categories of thought. What is very easy to understand is what it is stated there, and we'll review it in a moment in more detail. Cells divide. You know, you have one egg cell, divides into 2, into 4. So in this process, the DNA interacts with the environments. First, the environment within the egg, the environment within the mother's womb, then when a child is born, the environments where the child is exposed, food, family, later education, classmates, social environment, all of that. It's clear. It's very easy to understand that genes interact with the environment, and the outcome, therefore, is affected by the environment. If you understand that, try to incorporate it into your way of thinking, and if you were successful at the end of this lecture of doing that, you will be ahead of most experts. It's amazing how many experts, when they talk about these subjects, they know it in principle, they forget when they describe the consequences, the implications of these methodologies. So a few more words, a few more detail about each of these concepts. First, this is a scheme and outline of the double helical structure of DNA. I'm sure you have seen pictures like that. Each kind of atom is represented by a representative sphere of a different color. It's essentially a very simple molecule, DNA, but that's where the genetic information is contained. It's contained in the sequence of letters. This is a representation, simpler, of the DNA, where I have untwisted the two chains, the double helix, so that we can see the two strands separately. The genetic alphabet, the DNA alphabet, has only 4 letters, T, C, G, and A. Is in the sequence of these letters that the genetic information is contained. A human genome has 3 billion letters. There's a lot of letters and a lot of information. To give you an idea of what is implied here, if when this human genome was sequenced, one human genome was sequenced, that was announced just at the beginning of the millennium, more or less, if they would have printed the sequence of those letters in books, it would have required 500 volumes of the size of the Bible. Because that's not how we deal with this information. We deal with it electronically. We print it when we are dealing with short sequences, a few hundred, few thousand letters. But to deal with sequences much larger than that, we have to do it with, by means electronic. Now, very important is to realize that the information is contained in any one of the 2 strands. The 2 strands are complementary because of the very simple rule of the DNA that C, this molecule which is called C, can only pair with G, and T can only pair with A. So the two strands contain the same information. They are complementary. It's the same sequence, except that in every case you have the complementary letter. But that is where the genetic information is. This is what happens when the DNA replicates. The two strands unwind, and then each one of them serves as a negative template to synthesize a new strand. Wherever there is A in the original strand, there is going to be T. Now, if you look at the other strand, wherever there would have been T there, and now in the newly sequenced strand will be A. So the process, because of this strict rule of complementarity, A with T and G with C, is that one ends with two new DNA molecules which are identical to each other and identical to the parental molecule. That's what is the basic process that allows for the fidelity of biological reproduction and biological replication. Now, in this process, once in a while, a mutation happens. A mutation is when the wrong letter comes to be. And here I give you a simple example, but one that serves to illustrate the kinds of events that can happen and the consequences of them. First, let me point out that this now is in the alphabet of RNA rather than DNA. DNA is copied, so to speak, into RNA, and RNA goes into outside the nucleus and is synthesized proteins. And the proteins are the engines that run the life processes. And also they are one of the main components of our structures, of our bodies. Now, the DNA and the RNA are read 3 letters at a time. So the difference between RNA and DNA is that RNA, rather than having T, has U. So if we have these 3 letters, A, U, A, the amino acid, the components of proteins are called amino acids. And there are 20 of them that make up the proteins that are responsible for the functioning of living organisms. If you have this triplet, this sequence of 3 consecutive letters, A, U, A, the amino acid synthesizes isoleucine. Now, let us assume that the first letter is changed, that is, a mutation has occurred that affects the first letter. If it changes to G, the triplet now encodes a different amino acid called valine. It changes to C or to U, encodes an amino acid called leucine. Now, valine and leucine have very similar properties as isoleucine, so there's a very little biological consequence for the organism. Now, if you change the middle letter, depending on what letter is changed, now the synthesis will be for the amino acid arginine or lysine or threonine, and those are amino acids with very different properties. So that protein that is being synthesized by the gene of which these 3 letters are one of the triplets is going to be very impacted. And I'll give you in a moment an example. If the last letter is changed, in 2 cases you still have isoleucine. The amino acid encoded is the same as before, so no consequence. Now, this is a very, very different amino acid in properties, so that mutation that went to methionine will be of great consequence. So, one simple example of what happens with mutations. Hemoglobin. 98% of the hemoglobin that all of us have here, in children is different, and in babies and fetus is different, but for adult, and for this purpose you are adults, 98% of the hemoglobin consists of 4 chains, 2 of one kind called alpha and 2 another kind beta. The beta chain is a protein that consists of 146 amino acids. If one of them is changed, glutamic acid typically in most individuals changes to valine as represented there, then the gene, rather than coding for a normal beta hemoglobin chain, codes for a hemoglobin chain that results in a very severe form of anemia known as sickle cell anemia, because the red blood cells, rather than looking like donuts with a whole close but almost being very flat in the middle, they suddenly start to take the shape of a sickle. And then that does not function well, the individual cannot respire, cannot digest, and usually leads to death, typically by adolescence or by very early adulthood. And of course, the person suffers from many problems in the process. There's just a change of one letter, which has changed the 6 amino acid from glutamic acid to valine. That is a very dramatic mutation. Many genetic diseases arise by single mutations like this. Single mutations is when only one letter of the DNA is changed. There are other mutational processes that I will not go into the detail that also give rise to mutations, to diseases, to the genetic diseases. Now, the concept of development. Here you have to not only understand, and I will put it once more that way because you will understand it once I describe what I have represented in this figure, but then keep thinking about it every time that anybody tells you that they are going to clone a child or whether somebody should be cloned or not cloned. This is the notion, as specified earlier, that the individual is the result of the interaction of the DNA with the environment, where food comes, temperature, all sorts of processes are responses of the DNA to the environment and to other cells. Now, this is an experiment that was done here in California. I'm sure you have already noticed familiar names like Stanford, perhaps Mother, and Timberline. These were done in 3 experimental stations that the Carnegie Institution of Washington maintains in California. One very near the University of Stanford at sea level, 300 feet above sea level. One called Mother, just very near the entrance to Yosemite National Park, close to 5,000 feet. And the other one at Timberline, very much at the tree line, the point above which no vegetation can grow anymore. Now, cloning in plants is easy, in many plants, not all plants. This plant is called technically Potentilla glandulosa. The common name is cinquefoil, and it grows all over California from the foothills all the way up to tree line. The experiment that you will see that I'm going to describe are all based on the same process, which is cloning. Cloning in plants, as I was saying, in plants like this one is very easy because all what you have to do is to take 3 cuttings, take a plant, cut 3 little pieces, plant them wherever you want them to grow, and those are genetic clones one from the other. They come from the same individual, from the same plant. The new plants grow. So now the experiment. The experiment is that they have, in the first case, a clone or 3 clones from a plant from the Stanford garden. And one was planted in Stanford garden, one in the Mother station, and one in Timberline. 3 clones, genetically identical. This one does very well, as you can see here. The other one does well enough, but differently. The third one dies. So the genome, the genetic makeup of a plant from Stanford just cannot survive a tree line. Does that mean the tree line is the worst environment and Stanford is the best? Not necessarily. Look at the experiment on top. Here, the plant was collected in Timberline. Again, the same experiment. You take 3 pieces, and you plant one in Stanford, one in Mother, and one on Timberline. Now, look how skimpy the one from Stanford looks like. The one from Mother looks fairly prosperous. The one the one in Timberline also looks prosperous. It seems small, but actually it's healthier than the other 2, but you wouldn't know that from these photographs. And now a third experiment, 3 clones again, so genetically identical, but in this case, a plant that comes from Mother. So the one clone does reasonably well in the Stanford garden, does splendidly in mother, and that's rather poorly at timberline. So what are the implications of this? Two important implications is that cloning the genome of an individual does not mean you are going to have the same individual. That depends on the environment. The same clones may do very well or very badly depending on the environment. Second, and this in a way is the critical implication, is that there is no genome which is the best and there is no environment which is the best. You see, this genome is the best in this environment, but this does very poorly at timberline. On the other hand, the mother genome does very well in one environment, but does not so well in the other environment. There is no clear definition except for defects, which is what we'll be talking about very soon for genetic deficiencies, or in the case of humans, diseases. Except for genetic defects, there is no one. We look at the genome as a whole. There is no genome which is better and one which is worse, or an environment which is better or an environment which is worse. It depends. There are environments which are better or worse, and I will say something about that about humans. But in principle, there is no way to say which environment is the best. Depends on what genome you are talking about. So here you have something that I want you to keep in mind. Cloning is the duplication of the genes, not the duplication of the individual. The genes are cloned, the individual is not. And you can right away start to think of the implications of this for all these discussions about cloning. Now let me give you an example from animals. Animals cannot be cloned, certainly not easily and not successfully. But this experiment was done as follows. And for the moment, I'm going to describe what happens by referring to this line. These are rats. And the experimenters have a complex maze. And they put the rats one at a time in the maze. And there is food at the end. There are various ways to get to the end. And the rats smell the food, and they are hungry. So they try to get there as fast as they can, and they commit various errors. Once the rats have run once through the maze, they are run again. And then the experimenters were doing the following. The rats that made fewest errors, they were called bright rats, the smart guys. The ones that were making the most errors, they were called the dull rats, the stupid guys. And then in the next generation, you now keep them apart. You breed the bright rats on their own, but you run them again through the maze. And in the progeny, those who are the brightest, they are the ones who are selected to breed the following generation, and so on. So every generation, you take the brightest rats, and you use those to produce the following generation, and the following generation, and the following generation. And at the other end, in the other lineage, you take always the dullest rats, the ones who made most errors, and those are the ones you breed. And the next generation, you pick up the stupidest rats again and keep doing that. So now it's a test at the end of the experiment, more than 100 generations. Have they succeeded in changing the genetic makeup of these rats? Putting it differently, does the ability of rats to make errors or not make errors depends on the genes? The answer is yes. Look at what happens when these tests are done at the end. If the bright rats, when they are run through the maze, on the average they make about 115 errors. The stupid rats, on the average, make 165 errors, almost 50% more. This is experiment done in the same environment where they have been raised one generation after generation after generation. Just think about it for a moment in terms of IQ. If we are going to talk about, we would be talking about human beings, that will be like an IQ difference between 85 and 120. Quite a lot of difference. And it's genetic, it's reproducible. However, change the environment. You take rats of the same generation, but now they have been raised on the same strains, same selected lines, but raised under different conditions. So first, you raise them under restricted conditions. What is meant by that, I mean, you can think slums for humans. You know, the rats have not very good food, they're particularly crowded and the like. And now the average number of errors is 170, both identical. So these guys who were so smart in that environment, they suddenly become stupid in the other environment. The dull rats become a little stupider, but not much. But now the two are undifferentiated. It's not that they are not genetically different. It's that those genetic differences are not expressed in that defective environment. And again, you can think of the implications of this in terms of social environments, education, and the like. Now, what happens if you put them in a stimulating environment? You know, you give them the pleasure of living in some of these big mansions that we have around here on the hills, or you breed them and raise them and keep them in the Four Seasons Hotel, something like that. And what happens is that the rats, that presumably were very stupid and they were making errors, just by being raised in better environment, now they make about 120 errors. Just no more than the rats that have been selected for more than 100 generations to be smart. The bright rats improve, but very little. In fact, this difference at this end is not statistically significant. So when you put them in a very good environment, you take the rats from the slums and put them under favorable conditions, they suddenly act as much smarter. It's more than moving half age, as it were, at one point to another. Remember, these experiments are done by raising them from birth in a favorable environment or a typical environment or in an impoverished environment. So the same, see, you have the interaction between genes and environment. Genes affect, you know, the ability to go through a maze. Genes affect IQ, but that does not mean that the person who has higher IQ has better genes for IQ. It means that in the environment in which you are educated and you are being tested, you know, you do better than the other person. In another environment, you have been raised differently, might have been quite different. So back to the four techniques. Genetic counseling, genetic therapy, you remember the two techniques to correct diseases, to correct defects. Then germinal selection, which I will explain, and cloning for people who want to produce individuals with desirable traits, for example, my own traits by cloning myself, or simply improve certain qualities in humankind. I will discuss briefly one at a time. First, genetic counseling, and I already give you an example of a mutation that causes a severe genetic disease, sickle cell anemia. Sickle cell anemia is relatively frequent in certain human populations. In the United States, the frequency is not very high, considerably less than 1% or so of the frequency of the gene that causes sickle cell anemia. Now, whether genes cause disease or not depends on the following. For some genes, you have to have a defective gene inherited from the father and from the mother. For other genes, it's enough to inherit one of the genes defective, then the disease is expressed. Sickle cell anemia is of the first kind. So for an individual to suffer from this disease, has to inherit the sickle cell gene from the father and from the mother. The father and the mother are normal because they have one sickle cell gene and one normal gene each. And people who have one gene of each kind, the hemoglobin is okay, no problem. So they don't suffer from anemia. But if these individuals go into the hypothetical situation that I was mentioned earlier, they know there is sickle cell disease in their families, then sensible thing is that they go to the genetic counselor and say, you know, we want to know whether we are carriers. The expression is to be a carrier when you carry one of the genes defective, but you don't have the disease expressed because you need two for suffering from the disease. So they want to find out whether they are carriers. Well, there are relatively simple tests that have become available only in recent years, but they are relatively simple, can be ascertained. And assume for a moment that the counselor says, I'm sorry, you are both carriers. What next? Well, what is next is that they have 1 in 4 probability, 25% probability of having a child that who will suffer from sickle cell anemia. This is by the rules of assortment between genes and the gene cells. So 1 in 4 probability, 25% of having a child with a very severe disease is very high. So what will the prospective parents do? Can do many things. One say, well, I'll find another girlfriend and I will find another boyfriend, because so long as the other one is not a carrier, there will be no problem. The other one is, well, we will not have children, or we'll adopt children. The other one is, we'll take our chances. If they decide to take your chances, there is still one step further that they can do, which is what I call here, what is known as amniocentesis. This is a technique which is sampling amniotic fluid from the fetus, just through a needle through the belly of the mother. It's a relatively simple technique. It sounds horrible. You can imagine somebody going with a needle to the pregnant woman. But it's done relatively early, and it's a fairly simple technique and fairly safe. And then they can now look at these cells which are floating in the amniotic fluid, epithelial cells like skin cells, and find out whether the child is going to be normal or not. So now they find that the child is normal and fine. It's 3 out of 4 chances. What happens if they find that the child is defective? Well, there is the possibility of abortion. Will they abort or not? That's a personal decision. There are people, as you well know, who think that abortion is murder, and there are people who believe, and both of them are very decent people in the United States, and very large numbers of people. There are people who believe, on the contrary, that bringing a child to life, to birth, when with a very severe genetic defect, when that can be avoided, the death is the crime. The one has an obligation not to bring the child with such a defect to life. There are many variations of these themes depending on some diseases are more severe, some are less, some can be identified very early, some can be identified later. Some of them, whether the child has a certain defect or not, can be identified by ultrasound without even having to go through amniocentesis. But this is the purpose of all of this process, the genetic counseling, is trying to avoid, or at least inform prospective parents, to inform prospective parents about the possibilities and the consequences of having children with genetic defects. So it's informed, and ultimately is avoiding genetic diseases. Now let us go to the next technique, genetic therapy. This is high-tech genetics. This is what has become available starting about a dozen years ago or a little more. And this is, as I told you, imagine the gene for sickle cell anemia, for hemoglobin beta, and there is only one letter change. Well, you have, through genetic engineering, if you change that letter and put the correct one there in the DNA, the problem disappears. There are many ways of accomplishing that. One of them is trying to change the letter. The present technology does not allow to do that with humans. The other way is put a good gene next to the bad gene, and the good gene will produce good hemoglobin, even if some of the hemoglobin is no good. If there is enough good hemoglobin, that's fine. There is no anemia. And in this particular case, the process is to take cells from the marrow, the bone marrow, and then inject some genes of the appropriate kind, that is, the ones that code for a normal beta hemoglobin chain, and then reimplant the marrow and the bone marrow, and this person now is able to produce enough normal hemoglobin if the experiment has been successful, if the treatment has been successful. Produce enough hemoglobin now to be a normal individual. Good job. And a lot of this is being done now. However, it has to do with the fact that for every 2 successes that have been accomplished, there's at least 1 failure. And sometimes successes have become failures later. Let us now look at the other 2 kinds of therapies. The ones who try to produce better kinds of individuals, individuals with desirable traits, desirable in quotation marks, because it depends who desires them, or simply to improve mankind on the whole. As I told you, there was a Nobel laureate, no longer living, H.G. Muller, who is the one that proposed this technology. He said, let us take the sperm of very distinguished men, people of great accomplishments, whatever is the accomplishments, whatever are the accomplishments that we consider desirable, whether it's great political leadership or being a great artist or being very smart or a great mathematician, like most of you, I take it, are great mathematicians and great scientists. But he proposed to do that not at your age, but after years of life, when they would have proven that indeed they had a great, very successful life. He says, let us take the sperm and store it in banks, and that can be done in liquid nitrogen very easily. And then when a woman wants to conceive a child, rather than having sperm from her lover or her husband, her significant other, she could go with agreement with her husband to the sperm banks and take the sperm and say, I would like the sperm from, take a sample there, I like the guy, the one who accomplished so much as being a very distinguished mathematician, for example. There's the child I want to have. And then the genes of the individual, the father, if many women were to choose to do that, will be multiplied and they will spread, because conceivably that could be used for hundreds, thousands, tens of thousands, even millions. For sperm of a single man could produce millions of children, sufficient for it. Well, he said, that's not good enough. Women throughout their lifetimes produce 400 or 500 eggs, so we could flush them out. And in fact, can be flushed out relatively early age, very early age, and then store them, and women who are successful, then their eggs will make them available in an egg bank. And then now a couple who want to have a child said, I like, Marilyn Monroe, is an actress of my generation, not yours, eggs and Lenin sperm. Now, you laughed, but it's not there. It's not there by accident. It's there to make you laugh and to make you think about the problems, some of the problems that arise with that. Muller, as you can imagine, because these are examples of men that he said any woman would want to have a child fathered by sperm of one of these men. You can see what his political inclinations were, right? He was communist, and he was an American, but he was communist, and he thought, Lenin was one of the greatest human leaders. I should say, and you can laugh at it, but it is a way of making an important point, that he changed his political views later in life. And then when he gave examples of men whose sperm any woman would want to use to fertilize her eggs, Lenin was not there anymore. It comes to saying one obvious thing is that what qualities do you want in your children? Leonardo da Vinci, maybe he was a great artist. Lincoln was a great human leader. Lenin, no, probably for most of you. But more important is there is no way of telling. Remember the third concept of genetics? There is no way of telling that the genes of Lenin may in fact not give rise to a child that will be a great benefactor of mankind, of the genes of Lincoln that would give rise to a political villain. They just cannot predict that, because it's going to be impacted by the environment, and the environments are going to be different. I mean, Einstein and Hitler in different environments, Germany, one could have been the other. And there is no way in genetics or in anything else that we can predict that. Just to, I pressed that without meaning to, and I don't want you to be distracted from what I'm going to say, so I'll get back. One important piece of information for you. William Shockley is another Nobel laureate, but in this case, is the person who is a co-inventor of the transistor. As a consequence of his invention, he became very rich, and then he incorporated the sperm bank according to the desires of H. Muller and the prescriptions of H. Muller here in California. It was incorporated, supported with his funds. He endowed it. After, this was 15 years, maybe 20 years ago, after a dozen years or so, it was closed. The endowment had been exhausted. William Shockley, the donor, had died, and nobody seemed to have used that anybody knows. Nobody seems to have used the sperm bank. So that was the end of it. But some people were taking this seriously. Now, the reason why Muller was concerned about the genetic future of mankind is the following, and you can start reading if you want, or you can listen to me. You see, genetic therapy, or even normal medicine, an individual is born with a genetic defect, and through typical medicine or genetic therapy, we cure this individual. Now what happens? This individual who might have died otherwise before having children, now has children, and transmit the genes to the following generation. And there are many genetic diseases, and he thought that they will accumulate over the generations, and eventually we all become invalids, genetic invalids. You can read particularly what he says around the middle. He says that a point will come as a consequence of the use of medicine to cure genetic defects, where all we will be spending most of our life trying to doctor ourselves. We will put an artificial leg, something to take the place of your kidney, and so on, take medicine for other purposes, and we will have so many genetic defects, all of us. Why will the genetic defects accumulate? Because you have a process, a natural process is genetic defects appear by mutation. Genetic defects go out of the population by death of individuals who not leave progeny or leave fewer progeny. But now you cure this and they have children, then you have new mutations coming in and fewer or none going out. So the incidence of mutations, the frequency in the populations will increase. And he thought that in a few hundred years we will reach that situation. Well, he was a good geneticist, but he was not a good population geneticist. There's a branch of genetics, the one in which I work, and population genetics and evolution, where we study the dynamics of genes, how the gene changes, the genes change. And there is a simple formula among many others, but the one that I will use to make a point. This formula says the following: assume that the frequency of a defective gene is 1% in the whole population. You can think again, sickle cell gene. That is, the frequency is 1 divided by 100. If you cure all the individuals that carry the defect, which obviously is impossible, but let us assume that now we were to cure all individuals who are born with sickle cell anemia and they are able to have children. It will take the denominator in generations, it's 1 in 100, you remember? It will take 100 generations to double the frequency of the gene in the population. If you were to cure all sickle cell anemia people in the world, in the whole world, and successfully, it will take 100 generations, 3,000 years, to go from 1% to 2%. Well, it's not trivial, because there will be more children to be cured. But 3,000 years from now is a long way, the way the science is progressing. So his fear that something like this will happen was definitely excessive and completely out of place. The consequences of curing genetic diseases in terms of the effects on the population are very minor. They are not minor in terms of the individuals who are born with the disease. But even if we were to cure more and more genetic diseases, the impact in the population, the humankind genetic makeup, will be very small. Cloning cells, that consists of putting a single cell or a few in a petri dish in some appropriate container in the laboratory, and you give the right kind of food and stimulus to that cell and divides and multiplies. It has been done also for a long time It's not what we are talking about. We are talking about the cloning of individuals, or more precisely, in terms of what I was telling you earlier, the cloning of the genome of individuals, the genetic makeup of a full individual. Very different for plants than for animals. Plants, Mother Nature does it. Many plants reproduce by cloning. Farmers learned to do that hundreds, thousands of years ago. That is practically practiced normally. If a farmer wants to plant a vineyard and wants to plant a Cabernet Sauvignon vineyard, he goes to the nursery and he says, I want clone 73 Cabernet Sauvignon. That's the one I want to plant. And then the nursery will provide either with little plants that they have grown in the nursery, or simply cuttings of little pieces of branches of the vine and just plant them, and then he has cloned the same genome, same genetic makeup 50,000 times in a relatively small vineyard. So you have lots of clones, very different cloning mammals. The first successful case of cloning was Dolly, and I'm sure you have heard about this sheep. Was done in a laboratory in the UK, and was, 5, 6, 7 years ago, something like that, was hailed as a very important result, and with important, opening up important possibilities. Opens up very important possibilities in animal husbandry, because now if somebody has a dairy, say, for producing milk, has a big herd of cows for producing milk, he tries to have those cows that produce more milk to be the ones that produce also more babies. It's the ones that he wants to have reproduced, that he or she wants to reproduce, because he believes correctly that the ability to produce milk is determined by genes. He doesn't know what genes are involved, but a cow that produces more milk is likely to have children with more milk. The same is done to select bulls, although the method has to be a little more indirect. If one could clone the particular cow, which is the best at producing milk, at producing the most milk and the best milk, you just clone that cow as you clone 72 of Cabernet Sauvignon. Can it be done? Well, by now there have been at least 5 species of mammals that have been cloned: sheep, cows, pigs, rat, mice, rabbit. At least six that have been cloned. But there have been problems in all cases. There are individuals around who have, speaking of cloning themselves, this Dr. Severino Antinori in Rome, a gynecologist who has been practicing in vitro fertilization very successfully in Rome and is very highly recognized. And he and a scientist at the University of Kentucky in Lexington, both in dialogue with each other, announced that they were now to clone individuals, human individuals, genomes of human individuals. And in fact, at some point they announced that they have 2,000 women prepared and willing to be implanted with cloned eggs, and that some had already been implanted and were in development. Less serious people, the so-called Raelians, you may recall about 1 year ago, there was a young woman that appeared in television, was in all the evening news, in all the channels, who said that the Raelians, which is a religious cult, that they have already had a child born by cloning, and that you have 2 or 3 who were going to be born in the next few months. Almost everybody knowledgeable dismissed this claim at the time, and by now we know it had not happened. They simply don't have the means to do it, the technology, the laboratories, or the like. This is not something that can be done in a basement while nobody is watching. It requires very elaborate technology, aseptic conditions, what can be provided in a hospital. The Raelians, by the way, are people, it's called belief that the Earth was colonized by people coming from other planets and from other stars, and we descend all from these interplanetary migrants. But Antinori and Zavos are serious people, with recognized reputations. And that, when the announcement came, was very strange because there are many problems. And here you have some of the problems as pointed out by Ian Wilmot. Ian Wilmot is the person who directed the group that cloned Dolly. And you can read what it says. Dolly, they had 270 aborts before they have one success. So less than 0.5%. And successes have happened in other labs, mice, cattle, goats, pigs. Most of them die before or immediately after birth. No single cloned animal, as I was announcing earlier, has been perfectly normal. They suffer from immune deficiency, many of them, from organ failure. All of them tend to age very rapidly. That was one of the problems of Dolly. All of them have serious obesity problems, and they have to be treated with diets, special diets. Less than 1% or 1% say successes to birth, and then almost certain death or very severe defects there afterwards. Cloning human individual at this point in time, cloning the whole genome of a human individual, will be a crime. And indeed, as we'll see in a moment, is now considered a crime in many places. Moreover, the claims that are being made for cloning human individuals is, in some cases, people who are thinking of social engineering to clone lots of humans who are very intelligent, and then mankind will get to be smarter. I'd rather clone individuals who have higher standards of morality if I were to do some cloning rather than being smarter. But be that as it may, that doesn't make any sense. Remember that if you take an individual who has been very successful in whatever area of success you want to identify and is cloned, this new individual is going to live in a completely different environment. You really cannot predict what is going to come out. Remember the experiments with the cinquefoil. Now, human environments are considerably more complex. Familiar environment, the school environment, the social environment, politics, religion, all sorts of things. How these things are going to influence the development of the individual, you don't know. If I were so vain as to want to clone my genome, assuming that the technology improves, and it will improve. Eventually, I am anticipating that few years, whether it's 5 or 10 or 15, I don't know. But these problems will be corrected, and it will be possible to clone mammals successfully. When that comes, assume that I decide to clone my genes. Well, the child will be similar to me in eye color, blood group, and a few other things, probably overall stature and maybe makeup of the body. But all the things that count in life, the things that are important, character, preferences, personality, in all of that, there is no reason to think that it's going to be like me. It's going to be a completely different individual. It's going to be much more similar and much more influenced by children of his generation, or individuals of his generation, than by myself. So you cannot clone the individual. Remember the third genetic principle? You clone the genome of the individual, not the individual. Therefore, any desires to accomplish duplication of individuals are destined to failure. One cannot succeed as a question of basic principle. This is not a matter of technology to be improved. So very briefly, I reminding you of this experiment with the cinquefoil. Remember, the same genome in different environments, very different result. The same with the rats. It's better to try to improve the environment, for example, to improve the IQ of children, providing good schools and good food and good environment, social environment. It's probably more beneficial than trying to do it through the genes. So there is this genotype-environment interaction that was the third principle of genetics that you remember, and that becomes even much more important in humans because we are much more complex creatures. So keep this as the message of this lecture in practice when you talk to your friends or to your parents or people in the street who ask you, since you guys are so smart, you are students of the Cosmos program, what should I do about cloning? You cannot clone the individual. Okay? You can clone the genes, but not the individual. And in addition to biological problems and you cannot predict what is to come out, clearly the social implications of massive cloning are disastrous. So that will be a crime against mankind. And many people think that at least at present it should be a crime against mankind also to try to clone a single individual. Even when the technology becomes improved, there are many people that think that should not be done. Some people say, well, there may be circumstances under which an individual might want to clone himself or herself. I have given some thought to that possibility and explored a number of circumstances that have been mentioned or proposed by various people, and none of them can survive serious scrutiny. I think it's very difficult to find any kind of social, familiar, personal circumstance which will lead to the cloning of a whole genome of an individual, even when the techniques are improved. And indeed, very recent decisions throughout the world take this position. 3 weeks ago, the French Parliament adopted a new bioethics law that allows stem cell research, and there's a very important kind of research, and stated that human reproductive cloning is a crime against the human species. Reproductive cloning experiments will be punishable by up to 20 years in prison. And more recently, less than a week ago, the Japanese essentially passed a similar resolution, which now they are going to develop the operational implications. A policy that recommended that stem cells should be allowed, stem cell research, but not reproductive cloning. Now, stem cell research, and I will end by saying a few words about it, is a completely different kind of thing, because with the stem cell research, what is being done is taking embryonic cells and multiplying them in the laboratory so we can study them. Why stem cells? Why not other cells from my body? Because these are undifferentiated cells. The embryo has cells that can become nerve cells, muscle cells, kidney cells, eyes, you have it. All come from these embryo cells, these stem cells, before development. What we want to do is to learn, and I really don't work in this field experimentally, but what those who do, what they want to do is to learn what makes a cell to become a nerve cell and what makes it to become a muscle cell or a blood cell. Why is that important? For many reasons. Just the simplest one to explain, nerve cells. While if you get an injury and you cut yourself, or you have some skin that is taken off for some reason or another, the skin grows back. Nerve cells don't grow back. Consequence of that, two kinds of consequences. One that you have read about a lot in the media, Alzheimer's and Parkinson's disease. Very important diseases. Not for you, but yes, for people of my age. You know, some nerve cells degenerate and they cannot be repaired, they cannot be replaced. More important for everybody, in my point of view, because it affects mostly, or at least a lot of very young people, young meaning not near the age where people get Alzheimer's and Parkinson's, are injuries. There are lots of paraplegics and hemiplegics in wheelchairs, hundreds of thousands of them in the United States, millions throughout the world, as a consequence of accidents. Most of them, by the way, in the United States, car accidents, automobile accidents. What has happened is that they have, through whatever circumstances, they have had some cells damage, typical at the base of the head, because the head is big and the spinal cord comes from the brain down, and that has been broken. And now all what would need, one, is to have a few cells growing there and putting them back together, and that person could walk and run and be a normal person. But because nerve cells don't grow, these people cannot be cured, and they are condemned for all their lives to a wheelchair if scientists learn how to grow nerve cells, and so on for many other kinds of diseases. the benefits to mankind were enormous. Now, here, as you know, in the United States, we have a very polarized situation. There are many, including all scientists, specialists, knowledge about the subject, virtually all scientists, and even ethicists and so who are knowledgeable about the subject. The great majority think this research should be done. There are politicians and some others that they think this should not be done because these stem cells, they come from an embryo. An embryo is human life, so this should not be touched. Let me point out something to you that is not brought up in these discussions by those who are against stem cell research. They don't want embryos to be destroyed. Even if these embryos, they are nothing like a little human being. There's something more that looks like a small berry, like a raspberry, except it's the size of the pin, or the head of a pin. It's a tiny thing with few cells, maybe 60 cells or 120 cells, just all together, looking like a berry. That's how, after a few cell divisions, a human embryo looks. And this is the embryos that they want to use. Now, nobody is saying that we should produce embryos for this purpose. There are 400,000 of them available in freezers throughout the United States, in liquid nitrogen, conserved there. How come when in vitro fertilization is practiced, the typical processes that will induce the woman to overovulate, to hyperovulate, produce many eggs at the same time, the doctor collects them, fertilizes in vitro with the sperm of the father or whoever is going to be the donor, and then implants a few of them. If the implantation is successful, the other ones are put in a freezer. And they are there forever. If it is not successful, then implants a few more. But they don't want to have to go through the process of hyperovulation, inducing this ovulation again and again. So they do it once and fertilize many eggs, and they are ready. And they use 2 or 3 or 4 for implantation. The other ones are there. This is where you have, embryos are produced, which are going to be destroyed. Because what in the world is going to happen to these embryos? They're going to be destroyed or die or whatever you want to call it. Nobody's going to want to have an embryo which was produced, 10, 15, 20 years, or even 1 year ago, with the egg of some other woman and the sperm of somebody else to have it implanted in herself. She wants her own eggs. So these 400,000 eggs or so, which are estimated to be in freezers in the United States, will die for nothing. It will be very good for science, in my view. That's my conviction. I will not try to convince anybody who does not believe that for religious or moral reasons, but I'm expressing my convictions of this. If you want to avoid the destruction of human life, what the government should be doing is regulating in vitro fertilization so that not so many eggs are fertilized, so that not so many eggs end in these chambers eventually to die. But using some of these little raspberries the size of a head of a pin, who are already done there, who exist, just trying to cure human diseases, it seems to me a defensible policy, and certainly one that I happen to agree with. Thank you very much.